Yeah, hello everyone. My name is Julian Wiercinski. I'm not a PhD student of Alexandra's, even though the session might suggest it. Instead, I'm a postdoc at Harvard University in Edinburgh under Eric Gauger. And I want to talk about inferring energy transfer rate in complex systems, and most importantly, using experimental data. So rather from the bottom up, which is more like what you guys from UCL are thinking about, we want to kind of use experimental data and see how far we get. First of all, I want to acknowledge all of my colleagues and collaborators. So that's the quantum theory team in at Harvard University in Edinburgh. Special thanks also to my to the people who've contributed to the specific work. And then I also want to acknowledge the project that I'm employed by, which is the Apache project funded by the European Union. And I want to use this Apache project to also motivate why we might be interested in getting these rates from experimental data. And the Apache project, what it is about building, rather, not buying would be nice, a biomimetic sunlight pump laser. And the idea behind this is that if you look at the sun spectrum, it is super broadband, but the intensity is quite weak. And if you look at a typical lasing gain medium, so we see here neodymium, for example, you have incredibly low absorption and a very, very narrow lines. And this is exactly what we want if you want to have a very, very good lasing gain medium, but it's not what we want if you want to absorb as much sunlight as possible. So the idea is, what can we use? Well, we know that biology is incredible at absorbing sunlight. So the pitch would be to combine some biological components for absorbing sunlight with a great lasing gain medium to build a sunlight pump laser. And this is the general pitch, is you use something that you could act as a solar concentrator, like a chromatophore of purple bacteria. You attach some lasing gain medium. This could be a dye or maybe something like neodymium. And then you use the energy from the chromatophore to pump your lasing gain medium. And in the end, the goal of the project is to actually build a device, so also design the cavity structure and so on to make this thing work. And if you're more interested in the project, then please visit our project website later. So, and if we think about this step in which we assume we have absorbed the photon in our like solar concentrator medium, we want to transfer this energy onto our lasing gain medium. So one of the crucial aspects of this design is designing energy transport from our antenna that will collect our sunlight onto a lasing system. So this is just for reference, or just as an example, cyan and dye. And you can ask many, many questions about energy transport at this point. And we have heard about those questions before. So this is maybe the most immediate question is, what is the time that we need to transfer from our light harvesting antenna to our lasing system? Also, what is the steady state of the system? So how much of the energy in the end will end up in our lasing medium? What is the efficiency of the process? So how much will we lose in that process? And something that we heard a lot about in the last few talks is this transfer coherent or incoherent? So how much does coherence help or inhibit transport? But actually, that's a question I do not want to address. Instead, we are really interested in finding an estimate for the transfer time and the efficiency, and thereby also, of course, for the steady state. Just to maybe illustrate the challenge a bit better, actually, this is more how our system looks like. So we have a massive antenna comprised of heaps and heaps of molecules with a size that can range up to a micrometer. And we have lots and lots of possible dyes or lasing units that will be attached to this antenna. So if you want to kind of describe the system, of course, you need something that is super scalable. You can't use very, very sophisticated methods because they just don't have, they will just grow out of proportion. Instead, we want to find a way of finding a scalable method. And maybe to illustrate the point, I want to talk a little bit about how a quantum theorist like myself would approach this problem if I had all the choice in the world. I would say, well, I build a microscopic model for which I have a system Hamiltonian in which I have a number of sites with energies, and you have excitons that can hop between those sites. I would then have a Hamiltonian for the interaction of the system with the bath. That would be something like a collection of harmonic oscillators. And then I would have a spectral density or define a spectral density that captures the interaction of my system with the bath. And the spectral density in these biological systems can be super complex, right? And if in my dream scenario, I would know the spectral density. And we have in this conference already heard about some ways of solving these systems, and I want to plug here my collaborators very shamelessly. So for example, we heard about dumps, and also later on Adam will tell you about maybe a more scalable method of solving these systems. And of course, there are heaps of other methods around. We heard about HERM a lot. There's also T-Dopa tempo. There are other quantum master equations which can be employed. However, and I'm very, very grateful for all of the talks before, because I think they illustrated that there's lots of things you can do. We can be very accurate with our models. We can have all of the physical properties like trace preservation, like thermalization preserved, but doing all of this is incredibly hard. And it is work that requires the work of a PhD to just solve these intricate details. However, if you want to build something that solve a problem that is kind of big, or in which you don't have microscopic information, this is when you start kind of treading water a little bit, right? And just maybe because I was talking about these biological structures, there is actually lots of microscopic models for these structures around, right? But again, these are very, very inspiring papers by very, very smart people. And it took them lots of time and hard work to get to these results, right? And if you have an experimentalist breathing down your neck, asking you, what kind of dye do you want me to use? Then you don't have the time to solve this as intricately, right? So instead, how would we want to model a bio-inspired device? We will need some prior knowledge for this. So for example, the structure of the dyes, the structure of the light harvesting complexes that we want to use, and the electronic couplings between the sites, right? And the positions and the dipole moments of the acceptors. However, what we really don't want to do, because it's hard, is to fully characterize the environment spectral density, but instead rely on a set of limited experimental data. So for example, absorption and emission spectra. And if you have seen absorption and emission spectra from biological systems, they're also hard to get, right? And fairly noisy. So it's also hard to immediately infer the spectral density from these data. And what do you want to get out of this, right? We want to get out something that looks like a Pauli master equation, because it's just more scalable than a full quantum master equation. We want something that is sufficiently accurate, but we don't care about like percentages, right? And also we don't really, we want an estimate that is maybe on the conservative side. Importantly, we want all physical properties guaranteed. Right? So we want that our system thermalizes properly, that we get to the right steady states. And importantly, and I really want to drive home the point, is we don't want to have to go through all of the work of inferring all of the microscopic information for each component of our device. So how do we get to a model like this? And actually, quite unintuitively, I will start with a microscopic derivation, and then we will build our way up, and then we will see where we get. And the way how we do this, well, we start with a microscopic Hamiltonian with a bath. And then we choose the way of using a polaron master equation approach. So for everyone who's not familiar with this is a way of tackling strong system environment interaction by basically partially diagonalizing this, or by diagonalizing the system bath interaction. And what that physically means is that basically we absorb parts of the environmental vibrations into a displacement associated with the excited state. And this is a method that is very commonly used, for example, in solid state physics, but also many people in this audience will have worked with a polaron master equation. There are two main effects that you can get from this polaron master equation. So first, what you will see is that your Hamiltonian in the polaron frame will have reduced side-to-side coupling. So that reduces delocalization in your system. However, maybe as a comment, right, if you were to infer, or if you were to look at the absorption lines of biological systems, right, this renormalization would already be incorporated in the Hamiltonian that you would infer. And then also maybe more importantly, because this will lead us to where we want to get, is you get rates that lead to hopping between your eigenstates of this polaron frame Hamiltonian. And basically what we want to do is we want to really dissect these rates now and see if we can draw something more macroscopic out of those, right? And for this, I first want to go into one limit that is maybe very that many of you will know, which is the limit in which I assume that all of my dipoles are basically non-interacting on the level of the Hamiltonian. So this renormalization is so large that these interactions are suppressed. And then I can calculate the transfer rates between these individual sites, and I get an expression out of this. And if I massage this expression a little bit, I will find that this is exactly the limit in which you have first a resonant transfer. So this is effectively the spectral overlap between the emission spectrum of your donor and the absorption spectrum of your acceptor. So this is the Fred limit, and you very conveniently get it out of this polaron master equation. The other limit is a limit in which actually there are some clusters in this collection of dipoles. So our partition is more like this, right? And then we have an aggregate which acts as a donor and an aggregate that acts as an acceptor. And basically what you then can do is you can perform a separation of timescales assumption in which you say within this donor and acceptor, excitation will thermalize quickly before being transferred to the other aggregate. And then you can make a master equation out of this. And the idea of this master equation is basically if you have a spectrum of the donor emission and the acceptor absorption, you identify the lines of the corresponding to the eigenstates of the donor and the eigenstates of the acceptor. And you have then first the energy transfer from each eigenstate of the donor to each eigenstate of the acceptor. And this is the well-known multichromophoric Fred limit or generalized Fred theory that we have heard briefly mentioned before. Just for full transparency, let's look at these rates, these first rates, and they look horrendous, but we can again identify the same structure as for Fred. So we have a coupling between the eigenstates, which is which can be approximated by effectively the dipole-dipole coupling between these between the dipoles of the effective eigenstates, and we have something that looks like a spectral overlap. And the important question is that actually the spectral overlap of the eigenstates of the system or not? Right? So for this, you can derive an expression for the absorption spectrum by calculating the Fourier transform of the two-time correlation function. And what you see is, interestingly, the absorption line of each eigenstate is a convolution between what you can call a zero phonon line, which is effectively a Lorentzian line that is associated with the finite lifetime of your eigenstate in the polar master equation. And then you have the so-called phonon sidebands, which are associated with the absorption and emission of phonons. And these phonon sidebands, they are related actually to the hopping between eigenstates. So how we can interpret this physically is basically saying in MC fret, the transfer between eigenstate is facilitated by the absorption and emission of a phonon. And therefore, what is important for these rates is not the overall spectral overlap, but only the spectral overlap between the phonon sidebands. And of course, there's a question, and this is, let's say, I would say an open question yet, at least for us, is what is the contribution of the zero phonon line and the phonon sidebands to the overall spectrum? Right? Which one is broader, which one actually dominates the spectrum is a non-trivial question to ask. And importantly, I want to now draw your attention to one important thing that we were thinking about, which is the issue of thermalization. And for this, we will investigate these phonon sidebands a bit more. So if you think about these line shapes, you can effectively decompose your spectrum, which will look like the blue line, whereas the zero phonon line will look like the red one, and can decompose it into two parts. One half with a higher energy, and this is associated to a process in which you absorb a photon with a higher energy and then dissipate the excess energy into your bath by the emission of a phonon to reach the line or the two-level transition. Right? And this is a process that is proportional to n, but it's also spontaneously possible. Right? Even at zero temperature, this process exists. And then you have another part, which is the lower energy part of the absorption line. And this is a process where you first have to absorb energy from the bath to then make up the rest of the energy with your photon. And this is a process which of course relies on energy being in the bath. So this is a process that is suppressed at low temperatures. And this is why, importantly, every emission line of an eigenstate will be fundamentally asymmetric, with one part being suppressed. And interestingly, if you look at the Fret equations, you will show that the thermal balance in resonant energy transfer is encoded in exactly the spectral asymmetry. That means if I have an overlap between thermally consistent absorption and emission lines, all of my Fret rates will also lead to a thermalization of my whole system. So for us, it's important for our project is to use lines that are actually thermally consistent. However, you might have noticed that all of these lines still depend on the spectral density, which is a massive problem for us. So what we want to do, and I will go over this quickly, you can actually calculate in a high temperature limit and using a Drude-Lorentz spectral density assumption. You can calculate an quite complicated but analytic form of your phonon sidebands. And we can then now use this form, which has two fitting parameters, to fit experimental spectra. And then once we have these parameters, we can plug them back in to our first resonant energy transfer equations to make predictions about energy transfer. However, of course, and we have talked about this a little bit already, is that this is just an assumption of Drude-Lorentz spectral density. So this might not be physically the correct spectral density for the system, but because we are only interested in trends and not in the intricacies, we are completely fine with making this assumption. Let's now, in the last 2 minutes maybe, look at some applications of this. So these are preliminary results. But maybe just to illustrate the idea, I still want to present those. And for this, we look at a biological system, which is the purple bacteria. And we will look at the LH2 rings, which are the green rings here, and we will investigate transfer between those. And we will look at the LH1 complex with the reaction center in the middle and look at energy transfer between the LH1 and the reaction center. So first, for the LH2s, we can actually reproduce the experimental data in blue with our theory fairly well. Again, we are not looking for the most insane fit. We just want to reproduce the features well. And then we can use these parameters, plug them into our MC-FRET equations. And what we get here is at a distance of about 10 nanometers, we get transfer time, which is roughly consistent with literature. Again, these parameters, they are highly debatable. These spectra, you can find multiple parameter sets that fit. And again, because our underlying spectral density is an approximation, we do not necessarily want to compare this with literature. Similarly, we can look at LH1 and RC. We have data and we have a model that will not capture this peak. So this is by design. We see that we can fairly accurately, again, get the features of the reaction center absorption spectrum right. We can use the same parameters to kind of get the emission spectrum of the reaction center right. And we can then infer, oh, this is the emission spectrum. Oh, wait a sec. Yeah, so this is LH1, by the way. That's the absorption spectrum of LH1. We also can find parameters for this. And then we can plug in these parameters to make a prediction for the transfer rate. And these rates, at first, maybe don't instill the biggest confidence because they're a bit off from what we find in literature. However, there's lots of leeway still for us to optimize our fit. And secondly, I want to draw the attention to the fact that even though the numbers are maybe not 100% right yet, what we get is we get rates that are consistent with the thermal occupations that we will expect for the system to have based on the energy eigenstructure. And with that, I want to finish with a few conclusions. So again, a reminder about this cool project about building a biohybrid lasing device. We have talked about MC-FRET and how we can make it thermally consistent by using thermally consistent line shapes. And we have used this to fit some experimental data and make some preliminary predictions about energy transfer in these systems. Thank you very much.